Hello, BNG319 class. Welcome to the pre-lecture for our flipped classroom on Tuesday about combinatorial chemistry and drug design. But first, let's address a Muddiest point that we got from the November 16th class, talking about receptor-based drug design. Someone wanted to know if this process works with stereospecific enzymes / receptors. And does the new molecule or drug, in this case, does it alter the binding site somehow? So if we're thinking about stereospecific enzymes and receptors, and certainly the short answer is yes, it does work with this. And the thing is, you have stereospecific compounds that will end up being hits or potentially even lead compounds in your drug design and your drug discovery process. So you can have a racemic mixture that has like R and S things, S or D and L things in the mix. And if it's a stereospecific receptor or a stereospecific enzyme, you end up basically choosing bioactive or lead hits that have the configuration that fits with the specific enzyme or receptor. And does the new molecule alter the binding site? That's hard to say, and that's certainly a case-by-case thing. Usually not so much. The binding site is the binding site after all. And so what we are doing is we are looking at different compounds and different compounds that look like the ligand in question or the substrate in question. And we're looking at them not only for what types of groups they have attached to them, like hydrogen bond donors, acceptors, lipophilic parts, large parts, positively or negatively charged moieties on them, but we're also looking at them in 3D space because that becomes important as to how they're going to bind to the receptor or to the enzyme. So they need to have the right geometry, the right 3D geometry, in order to be able to accomplish that. And so if there are stereospecific parts, they need to be present, and they need to be in the right stereospecificity. Otherwise, it's not going to work. And so we have binding site models that we can make, especially if we have the receptor or the enzyme, the 3D structure, the crystal structure of these things. We can look and see how the enzyme fits in, like where does it bind, with what amino acids does it interact. So if we have a ligand or a potential drug that has, say, one group facing the wrong direction, we might not be able to make enough contact with the key amino acids in the binding site. And certainly if that keeps key residues in the catalytic site away from the actual substrate molecule or drug molecule in this case, then said drug won't be able to function. Won't be able to function as a drug. It may not even bind to the receptor or to the enzyme. And one example of stereospecificity in drugs is this warfarin, where we have either R or S warfarin. And these are drugs that have been used as anticoagulants. And there's variations in response to warfarin, and that results from variability in the drug's metabolism and how we metabolize the drug. Different warfarin reduction kinetics, and the first step in metabolizing warfarin is the reduction of warfarin at a certain spot. The different kinetics depend on that stereochemistry. And now this is a case not necessarily where one drug will bind and the other won't. This is about different metabolic rates and consequently different enzyme activities, different Michaelis-Menten kinetics in this case. So, and this depends on the stereospecificity. You can see this is the alpha carbon that we're talking about. This is the chiral carbon that we're talking about, where we have this phenyl group and the hydrogen. And the R form looks like this, and the S form looks like this. And for the R form, the Vmax is higher than for the S form, and the Km is lower for the R form than it is for the S form. And so this ends up being key if we think about the variations in response to warfarin. So we have two different stereoisomers here that have two different kinetics in terms of how they are metabolized. And again, the start of this process is the reduction of the molecule at a certain region. Another example is ibuprofen. Ibuprofen is a COX inhibitor. COX is cyclooxygenase, which oxygenates arachidonic acid and endocannabinoids, and it basically acts as a pain reliever. Obviously, we know that ibuprofen acts as a pain reliever. We've probably been taking it for several decades. And so these COX-2 inhibitors, they non-specifically block the substrate binding site in the COX enzyme. And they basically act, even though they're non-specifically blocking, they're acting as competitive inhibitors because it's at the active site. Thus, they act as pain relievers here. And then so we can see arachidonic acid would like to try to bind, but it is unable to because ibuprofen is blocking that active site. And so only the S form of ibuprofen is active. And you can see here the difference between the S and the R form. And again, you see the Fisher projections, and this is the chiral carbon in question. So only the S form is able to inhibit COX-2 activity. So it's basically in the right configuration to be able to non-specifically block the site. Any other way, and it is not able to do that. So this R version is not able to inhibit in that manner. Okay, so let's get on to what we are going to be doing on Tuesday. We're going to be doing a little exercise in combinatorial chemistry. What is that? What are we talking about here? And so combinatorial chemistry on the large scale and on the typical industrial scale is the synthesis of millions of molecules that are hopeful drugs and the subsequent test of those molecules to see what their biological activity is, to see if they inhibit key enzymes, if they inhibit key drug targets or have some effect, some biological effect on key drug targets. This is usually done, in fact this is almost always done in a high throughput fashion because we are testing so many different molecules. So you always have robots that dispense the different compounds and different assay mixtures, so we can see exactly what these compounds are capable of doing. Are they able to inhibit enzyme activity? Are they able to bind to active sites in receptors or bind to as ligands to receptors and act as either agonists or antagonists? Depends on what we're trying to do. But doing millions and millions of molecules in a high throughput fashion is able to decrease that discovery to market time that we talked about last week. So I showed you a graphic that indicated that discovery to market time was roughly anywhere between 10 to 14 years. This type of combinatorial chemistry approach could decrease that time to maybe 5 to 8 years. And the key for this is, and this is what kind of the hallmark of combinatorial chemistry, the key for this is using mixtures of compounds, so combination of components and kind of reacting them together to make a mega library of different molecules that could potentially be used as active compounds, as drugs. And so we can take a look at this in a vague fashion here when we look at combinatorial chemistry versus traditional organic synthesis approaches. So with our traditional organic synthesis that we talked about maybe a month or two ago, we see, and we have A, we have B, we react the two together, we yield C. And that's great. But suppose we had kind of a basket of different types of A's, that have one specific functional group, and a bunch of different B's that have another specific functional group, and the two functional groups are able to react with each other. So we can then make a whole pool of C's in a variety of combinations. So, A1 can react with B2, A1 can react also with B1, A2 can react with B2, B1, and B3, etc., 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 to make many combinations of molecules in that way. And so then we're left with a library of C-type compounds and hopefully these C-type compounds are of the type that could potentially be used in looking for some kind of drug or other bioactive molecule. And here's kind of a very basic illustration where you have maybe a scaffold. It doesn't have to be as simple as an amide bond, but it can be. And we have three locations where we can react some sort of moiety onto the scaffold. The red location, the blue location, and the green location. And we have these libraries of different small molecules that can react with the scaffold in the specific places. So we have a bunch of different shaped reds, we have a bunch of different shaped blues, and we have a bunch of different shaped greens, and we can get a variety of combinations of these things to make a big library of combinatorial molecules that we can then test for their activity. And here's a more real example where we have this BZA molecule, this benzodiazepine molecule. And so we're starting with this as a scaffold. And we're starting with this as a scaffold. We have different spots where we can react or put different moieties onto. We have like the R1 spot, R2, R3, and R4. And we have different things that can go in each of these spots. So we can react those things and stick all that stuff somewhere onto the BZA molecule and come up with a bunch of specific structures attached to BZA. So now they have all these different R groups, and hopefully we can use this library of BZA type compounds to see which is able to perhaps interact with the drug target. And so when we think about what we're doing here in making all this stuff, we can go fishing with a net that's huge, a net that's the size of our state, the state of Massachusetts. But what do we do with it? If we are trying to catch fish, we'd have better luck if we toss it over the Atlantic Ocean than, say, if we threw it over the Rocky Mountains. So what does that mean for us? Well, what that's saying is that it's better to start with a general structure. So you're not really just looking for a needle in a haystack. You're starting with a general structure that has some sort of efficacy or some sort of relation to the either to the receptor or to the enzyme that you're looking at, or even to the ligand or substrate that you're kind of modeling it off of, depending on what avenue you're coming at it from. So it's best to start with that general structure. So this BZA presumably can interact with certain receptors or certain enzymes. And we add all this other stuff onto it in hopes of making a BZA-like molecule that can have some sort of bioactivity. And so the advantages when we compare, again, our conventional strategy versus the combinatorial strategy that we're talking about here, conventional strategy is one molecule at a time, kind of like we showed in a few slides ago. The combinatorial strategy is many molecules at a time. So you're making things a lot faster. And for the conventional strategy, you make the molecule, you purify it, and then you test it. Whereas for the combinatorial strategy, and this is going to be key for some of the questions on the flipped class activity assignment, you'll make molecules, you'll test and see if the group of molecules has some sort of biological activity that you're looking for, and then when you've kind of narrowed it down, based on a number of different combinatorial libraries, you can then purify out of that specific batch the actual bioactive molecule. So you can test millions at a time, but in the end you can just take a smaller batch, and pull that bioactive molecule out of there. Conventional, you maybe do hundreds of molecules in a month, whereas combinatorial, you can test thousands of molecules in a month, maybe even more. Depends on how good your robots are. With conventional, and again you can probably see a theme here, slower lead generation. Combinatorial, just because of the sheer bulk of it all, faster lead generation. Higher risk of failure with your conventional method, a lower risk of failure with combinatorial, because you have, again, so many different compounds that you've made, and with that you can identify the lead. And so what we're going to look at are these molecules called hydrazines, and a product of reaction of hydrazines and something else, hydrazones. And so what you can see here, these are hydrazines. This is the simplest hydrazine right here. Again, you've got the N-N bond here, and you've got the rest of the bonds are taken up by hydrogens. But the generic hydrazine structure looks like this. So you can have a bunch of R groups in place of these H's, but you basically have the N-N bond here. Hydrazones look like this. So again, we have an R group. We have a nitrogen double bonded to the carbon here, and we can have either a hydrogen here, or we can have another R group here. But we always have a nitrogen bonded here, double bonded to the carbon, plus the nitrogen-nitrogen bond here. And so how do we form hydrazone molecules from hydrazines? Well, we can look at the action of hydrazines on either aldehydes or ketones. So we have an aldehyde or a ketone here, we have a hydrazine. Essentially what we end up with is the hydrazone right here. And you can see that this is a condensation reaction, so we're getting H2O as a co-product here when we make hydrazones. And here's that kind of shown a little bit in more graphic here, where again, this is the water we're removing, and then we end up with a hydrazone plus water here. So we have an acetaldehyde, hydrazine, we make the acetaldehyde hydrazone plus some water. And so for you mechanism buffs, here's what that looks like. Our electron donor here, the nitrogen, is going to donate these 2 electrons to the electrophile, our friend the carbonyl carbon. We move some electrons up to the oxygen. We have a negatively charged oxygen. It can then pull this hydrogen off, and we have some movement of electrons from bonds to the H, and then to the H bond to the nitrogen to this bond here, and we end up with the hydrazone. So what you're going to be doing is on-paper combinatorial chemistry. Are we going to do millions of compounds? No, we're not. We'll do about 9 compounds, and that is going to be more than enough. So we have an active compound library where you'll have, on one end, you'll have either an aldehyde. I think it looks like we have all aldehydes here, things with aldehyde group, and then a bunch of things in the B column that have a hydrazine on them, some kind of hydrazine group. So you will make these hydrazones, and so you can see how these might come into being, and kind of where, here's B1 is here. You can see where it makes the hydrazine here. A1 here, A2 there, A3. And so you can see how they fit together, kind of like a puzzle piece, to make a bunch of different hydrazones. And what's the key with the hydrazones? Well, they're going to fit into some kind of drug target. So we've made up a drug target. Perhaps this is an enzyme active site. Perhaps this is something, it's a part of a receptor or something like that. But it's got key amino acids. It's got some hydroxyls here. Presumably these are serines or something like that. And we've got a charged amino acid residue here and a hydrophobic amino acid here. So you will try to fit the hydrazones that you've made into this active site to try to figure out what is the best fit. And so you're going to fit the lead compound into the enzyme active site, or whatever you want to call it. And so you'll do on-paper combinatorial chemistry. I've assigned groups of, I think, 5 to 6, actually. So you, as a group, will work together to answer the questions. And I'll hand out a packet on Tuesday, and everyone will turn in their individual attempts as an assignment for credit. And so here are the group assignments. So you want to get into your groups first thing on Tuesday. I will, after this video is finished downloading or uploading, I will post it to my courses, so hopefully you should have everything at that point, and you can review all this stuff. Please watch this video. Obviously you have watched it if you're listening to me say that right now, so that doesn't apply to you actually. But anyway, hopefully everyone will watch this video and kind of have an idea of what they're going to do when we get in Tuesday, and we shouldn't waste any time in getting going, and hopefully you can get a good deal of it done. And I will be milling around the classroom as always, and I'll do my best to help you if you get stuck on any portion of this assignment. So I'll see you on Tuesday, and good luck.